a very warm introduction.
Thank you.
Sylvain is fairly recently, but
Yeah.
following things from the distance for a long time. He's a professor at ETS in doing a lot of tools and infrastructure for neuroimaging with application to health, brain health, and psychiatry and others. The particular angle, which I think is really interesting, and is the openness as well of the tools, but also pushing for inclusivity and making sure that those tools and those methods are well adapted to the whole population. I think that's one of the aspects that was striking when you look at what Sylvain does. You had a long career in the US before coming to Montreal, so it's just this long career has been extremely productive, and now as well, productivity is amazing. And I think to me, what makes me close as well to Sylvain in some sense is this aspect that science is often on the side of nice results on the brain and aspect, but the whole work to actually make that feasible and easy and reproducible and open, through infrastructure and tools is really one of the things that makes our science much more efficient and so I think that's one of the things that you're pursuing, and that I think it's so commendable. Thank you, Sylvain.
Thank you. Well, thank you for having me and inviting me. So before I start, because I think I've been told it's quite diverse crowd here. So how many of you have analyzed MRI data? How many of you have developed tools to process MRI data? Okay, a few. Okay. And so how many of you were looking at the brain? Okay, so that gives me a bit of an idea of what we'll talk about today and how much in depth that will go. So all the work I'm going to be presenting is primarily the work of my students and engineers and research assistants. Here's a snapshot of the projects they're working on. Before I start, I need to acknowledge all of the funding that helped all the work that you'll see presented. The chair, some NIMH funding, and a discovery grant. So we'll talk about today about two main themes. One is neuroimaging and how do you develop tools to better process MR images, and what I think today some of the challenges we can have on things that often people don't think about, and they think, I run my pipeline, I have cortical thickness or brain volumes, I'm good to go. Well, it's a little bit more complex than this. And then the second theme, which I think is important for this crowd. And so here, how many of you here consider yourselves data scientists? Okay, how many here consider yourself data engineers? Anyone has an idea of what's the difference between a data engineer and a data scientist? No. Okay, so we'll talk a little bit about this, because what is important, what I want to talk about here is that as a data scientist, you're going to get this wonderful database or spreadsheet from which you can run your amazing R code. But the work that comes between the moment somebody is interviewed in a room and that data is available to you takes a lot of work, and I'll go over some of the themes on how we can do that. So this is the two theme of my research. I'll have a little snippet about how, in my view, open and reproducible science is critical. And I won't talk too much about application. I'll talk a little bit about schizophrenia, but I've worked in schizophrenia, brain injury, and other disorders, repetitive head impact. So let's talk about the first thing. For those of you who have played with MRI data, how many of you have done quality checking on MRI data? Okay, a few of you. So MRI data, just like a regular picture, is messy, it's slow. Typically, you spend anywhere between 10 minutes and over an hour in a scanner. A single image or 3D volume of your brain typically takes 5 minutes, or even though now we can go much, much faster. And what that means, you have highly sensitive equipment, high Tesla, magnetic field, and it's susceptible to many, many different kinds of artifacts. And here are just some examples. The top one is what happens if you move your head in the scanner. So here you see a nice image where there's very little motion. And then here somebody moved a lot, and you see kind of like these concentric lines and blurry images. Then you have other things that can happen in the scanner, such as ghosting. If you have a lot of blood flow, that can create some artifact, some noise, some errors with the hardware that you can find. So part of the work we do in my lab is to try to mitigate for those problems so we have nicer images. And I'll only talk today about the work that Charlie's doing, because I think it's important to know that these kind of things have an impact on the data that you analyze in the end. And sometimes an impact that you cannot really see well until you measure it. So here's his motion, very little motion, or close to no motion observable by the eye, right? And then all the way to, if you're a research assistant in the lab and you have to QC, you would say, okay, this is a 1, it's a great scan, and this is a 4. Has too much motion. I can't really run my pipeline on this. So I'm going to discard these cases and call the site and say, please rescan this participant because the data is not good enough. And then, but even if you stay within this little area of the 1s and 2s, and what you see here in the x-axis is the amount of motion that we're able to measure. Okay? So here is no motion and some level of motion. This is kind of an average measure over a certain amount of time, how much you move your head. And what you see here is an estimation of the cortical thickness of the left hemisphere of the brain. So what that means is that if I move my head in the scanner, even by a little bit, my cortical thickness estimation is reduced. My cortical thickness biologically has not changed. It's not because I move my head that my cortex gets thinner. But the tools we use will estimate a lower number. So it's pretty critical that we can estimate this number before you run an analysis, especially if you look at, want to do group differences between a population that typically does not have a lot of motion. The stereotype would be ADHD versus normal kids, okay? Are you measuring the biological differences, or are you just measuring the fact that they move more in the scan? So up to today, there's no tool that given this image or that image gives you that number. So that's what we've been working on. And the way we've done this is by simulating motion and then trying to predict it. I won't give you all of the details, but of course, it's a deep learning network. But the crux of it is that, if we synthetically, if you take a clean image and you introduce synthetic motion to this image, then you can train a neural network to estimate that motion. We can show that we do pretty well, even though the more motion, the more variance we have. And what's very important is all of this is trained on synthetic data, but when you apply it to standard data sets that we find in the literature, the Healthy Brain Network, the Human Connectome Project in Early Psychosis, and others, we find this trend. And so for these data sets, it's important to have this number and incorporate it in the statistical analysis. And so something to be aware of, in particular for certain sites, and this is kids, this is older adults, and this is the HCP young adult data, which has one of the most stringent protocol for scanning people in the highest quality control, and you can even see if there's less of a trend. So this is this first part about data quality and how important it is to look at it and see that it may have repercussion way down the line, even though it doesn't necessarily appear on the scan. So the second aspect that I want to talk a little bit in neuroimaging is this concept of computational morphometry. Has anyone heard about phrenology? Yeah, what is phrenology? Okay.
It's the study of how skull shape determines your behavioral.
Okay. So this is exactly this, right? You have these measuring tool, you have these ideas that this is where things are, and then the bigger it is, the better or the worse you are at certain things, right? And so, of course, this is not founded in any sort of reliable science, But what we do with brain MRIs to a certain extent is very similar to this, and this is what computational morphometry is. We look at the quantitative measurement of the form of certain objects in your brain, and from this we infer certain behavior or differences between populations. So it's really important because the size and the shape of structures can be proxy measures for underlying biological mechanism. And the main measures that are used are usually the volume of regions of interest or the thickness of the cortex. Those are the two primary measures that are used. And so, how many of you here have heard about segmentation, image segmentation? Okay, quite a few. So I don't have to go too much into this, but this is what we get. This is a 2:1 weighted image, and this is what we want. So we can look at these regions independently, create tables, analyze the volumes over populations or regression or prediction, whatever you want to do. This process cannot be done without some a priori knowledge. You cannot just look at the contrast and say, you know, this structure is whatever, this structure is the hippocampus. You need to have some a priori spatial information in the form of atlases or training data. And so that means that a neuroanatomist is looking at some histology atlas, ex vivo atlas, and knows the different cells and is going to look at this image and say, okay, this is this area in the brain and manually outline it. And once you have enough training data, you can train algorithms or build probabilistic atlases to generate these automatic tools to do this. The problem today is the current atlases or the training data sets are really limited in the number of studies. So the most commonly used tool to do brain segmentation is a traditional tool called FreeSurfer. Anyone knows FreeSurfer here? A few people, okay? And it relies on 40 brains. Okay? So it was trained with 40 brains, and all of the information you get from all of these images, in 99% of all the neuroimaging morphometric studies come from these 40 brains that were annotated. Okay? They're limited demographic representation, and so FreeSurfer is actually not that bad in terms of displaying male, female, a decent age range, but if you want to look at other kinds of demographics, forget. And then it's based also on all neuroanatomical definitions. So, you know, FreeSurfer was built in 1990s, early 2000. The resolution of the scans was not what it is today. It was 1.5 plus millimeter cube. And so today we get sub-millimeter accuracy. We've learned about the anatomy through histological study. And so there are new rules and new structures that we can outline. And so this is part of my work, and it's frankly not something that's going to make the cover of Nature, but we build these tools, right, to draw new brains, and we try to make it as efficient and as simple for the neuroanatomist to use. And so here's the setup. We work with somebody at Boston University. He has a keyboard with shortcuts. He can outline with a stylus. He has a big screen, and it has a lot of shortcuts, so he can go through these brains as quickly as possible. And now we're working on semi-automated tools because you have to place landmarks. There's lots of things you have to do. Another thing that we do is, for those of you who are familiar with diffusion MRI, in diffusion MRI, you extract these tracts in 3D that represent the white matter of the brain. And if you want to bundle them together, annotate them, it's very difficult to do this by looking at a 2D screen because you don't have any depth perception. And so we're doing some VR, meta helmets where you can actually inspect and edit these tracks using sort of modern tools to do this. Okay, why do this? I mean, I've said we need more training data, but does that really matter in the end? And so I have a couple slides to show you some results that say I think it does. So this is manually annotated data in the same data set versus FreeSurfer estimated data. We have a few structures, and you can see that, well, there are differences. There's a bias. And that is the FreeSurfer tends to estimate certain structure quite a bit larger than the neuroanatomist does. What's a little bit concerning is if we look at this side, here we're looking at asymmetry. Is the left part of your brain bigger than the right part of your brain in certain structures? And then what we find is there's different results, right? And so here, okay, for the putamen, free surfer and HOA, so the manual will tell you it's about the same asymmetry. But in here, free surfer tells you there is a strong asymmetry, but the manual rater does not find this asymmetry. So there's something about manual tracing that is different and that could have an impact when you do your analysis. Another thing that's important in what JB mentioned is this concept of bias. So, today, if you run FreeSurfer or any deep learning tool, it's really hard to know if it performs better in a certain population than another population. If you take an extreme case where, okay, I am going to train my deep learning model on individuals between the age of 18 and 22 who are francophone, raised in Montreal, okay? If I do that, is my segmentation tool going to work just as well when I go to, I don't know, South Africa population? How do I know that? And in some cases, it may not matter. The problem is we don't know. We don't have a ground truth. And so this is work that we've done here where we tested a little bit these extremes. So here's a manual segmentation. Okay, and this is the average volume for one particular structure. Okay. So over a population of about 100 participants, we get this spread. And then the exercise that we did is each color represents a method. So here the method is called ANT, which is a traditional tool. Here you have COTR, which is a transformer-based deep learning tool, and then UNet, which is probably the most famous UNet segmentation tool for medical imaging. And here's UNES, which is a transformer version of UNet. And then each independent axis here is that methodology trained with a very extreme bias. So here you have well, it's better if I say here, you have a model trained only with black females. Okay? All the training data are black females. Here, all the training data are black males, etc. Okay? And then we compare over the entire population, are there any differences kind of in these volumes? So you can see, for example, for NN UNet, it doesn't seem to make a big difference what you train it with. Even at the most extreme level, the volumes are about the same. For the traditional method like ANT, well, it's actually quite biased in certain scenarios. So is UNEST, even though it's less apparent. So be careful what you train your data with, but not always, right? And so you need to take care of those things to make sure, because of course you want to annotate the least amount of data possible with the data that's available to you. So, you don't want to go and have somebody manually trace 100,000 brains so you have the entire world population represented, but you need to be careful that you may have some biases. So the last point I want to make in this sort of neuroimaging chapter is this cortical surface reconstruction. So there's this sort of two primary representation when we do morphometric analysis for MRI. And in neuroimaging, one is a volume-based, voxel-based. So, every little point here is a pixel or voxel in 3D. If you add them up, that'll tell you the size of a structure. If you want to look at things beyond volume, it's good to go to a mesh representation, especially for the cortex. That allows you to calculate the thickness, which you can shoot, rays from the white matter to the gray matter edge. You can look at the curvature, and all of these things are important features for analyses, and they're much easier to extract if you have a mesh surface-based representation than if you have a voxel-based representation. We've looked a little bit at this, and one of the issues with the current methods, not just deep learning, is they don't really pay attention to topological features the way they should. So what can happen is, for example, the left hemisphere enters the right hemisphere. Biologically, that's not really possible. And then when you deal with mesh, you have these tiny triangles, and when you fold in these really tiny gyri, they can fold onto themselves and create things that are not really great. So we developed this deep learning tool that does this prediction in a combined way. And I don't know if you can see this, but the blue and the kind of blue is this traditional method that shows this intersection, and what we can get with the yellow is they get very close, but they don't actually overlap. So we think this is important to try to mimic the biology as much as possible. So I'll conclude this first part. I think, my take-home message is morphometry remains a key methodology for understanding the brain. We need new reference training data to train the next generation set of tools, and we should take special care to limit biases, whether it's technological, like this motion bias that we had, or demographics, racial categories or ethnic backgrounds or whatever. So I'm going to shift a little bit because, up until now, all I've presented more or less is consuming data, which we all do. But where does this data come from? How does it get in our laptop? And so I think this is something few people talk about, very few works are published, but that black box needs to be opened a little bit and presented. And so the example I want to give you here is the work I've been doing as part of this large initiative called the Accelerating Medicine Partnership in Schizophrenia. And the goal is to better understand clinical high risk for psychosis. So anybody here, does everyone know here what schizophrenia is? Who does not know what schizophrenia is? Okay. So what we're interested in this project is to look at people who are increased clinical risk of having a psychotic episode. So we're talking about teenagers, early adults who have symptoms. They may have already hallucinations, they may have some of the negative symptoms like withdrawal, etc. But they haven't really, these symptoms haven't been severe enough that they actually require a first hospitalization or full psychotic episode where you disconnect from reality for a little bit. And so there's a lot of work in trying to understand, because not everyone who has these symptoms will convert to full psychosis. The number of people who convert to psychosis, the verdict is still out. Some people say 10%, some people say as much as 35%, but for the sake of argument today, we'll say about 25% of people who have symptoms will convert to full psychosis. About half of them will get better, and the rest will neither get worse or better. They'll just go through this lifetime of attenuated symptoms on this journey. So there's no size-fit-all for CHR intervention, and so we need new treatment. We need to better understand. We need to know, and especially who is going to convert. Can we build a prediction algorithm, a risk calculator? And so, as you know, if you're in this program, if we want to do this well, we need big multimodal data. And this is what NIMH, so the National Institute of Mental Health, in the US, and some private partners funded this very large effort to try to recruit individuals that are at risk of psychosis across the world, so we would have a diverse representation from different cultural backgrounds, and try to tease apart what are the factors that lead to psychosis, and if we know those factors, can we link them to certain biological markers that then drug companies can target for the treatment. So this is the setup. It's a $100 million effort. The goal is to recruit about 2,500 participants, the age 12 to 30. There's 42 sites around the world. Longitudinal design for a period of 2 years, wide range of domains. So here's the setup. You have a 2-year time frame. You do a lot of the, you have pretty much everything you can think of, clinical questionnaire, neurocognitive tests, MRI, EEG, fluid biomarker, both saliva and blood, actigraphy. They were the kind of a Fitbit watch type thing. They have an app on their phone that tracks GPS, that has daily surveys. What else? They do audio recording and Zoom recording, so we can check also facial expression and different pose estimate and see how people interact, eye gaze, all of that stuff. So all of this is collected across the world, and with the goal of saying, if I take all of this information, can I predict outcome? Do I need a little bit of longitudinal data to say that this person is going to be doing course today? And really my role in this was to fill in the question mark. Okay. We have 43 acquisition sites. Each person is seeing X number of patients. They have to acquire all of this data. And you could be in Santiago, Chile, or in Hong Kong, or in Montreal. McGill is one of the participating sites. And ultimately, we want to provide a curated release of all of this data for you, the research community, to look at it. And when I say you, the research community, anyone here who goes through the credentialing process can have access to this data. In fact, no one who's part of the project can publish anything that's not in here. So you can't play around with data that is not available to the rest of the world. And so what does that mean, opening this box, this question mark? Well, we're talking about data management, processing, and archival, infrastructure, pipelines, to make sure you don't reveal the name of participants. If you want to analyze GPS data, it means we know where you are within a few meters every few milliseconds. That data is something you need to be careful with. Is the data complete? We need to do quality checks, we need to do frequent data sharing, all of this stuff. So how do you go about doing this? So anybody here who's worked in the software engineering world? Okay. Does agile mean something to you? Okay. Does DevOps mean something to you? Okay. What are these principles? What are they trying to achieve?
I guess just moving data around, storing data, the whole pipeline.
From a software standpoint, what's agile, what's DevOps? What are people trying to do?
Oh, to do small increments of work. I guess.
Yeah. So okay. We're in the 80s and 90s. The primary model is waterfall. Okay? We write a 25-page requirement document. We spend 2 years coding it. We give it to the consumer. They don't like it. Okay? There's many examples in 2025 where that happened. We can talk about recent government work here. But so agile DevOps is kind of a response to this. Minimal viable product. Let's try to get something in the hands of people as quickly as possible, as safely as possible, so we get the feedback, right? So you do scrums, you do sprints, you do all of these CAMBAM, all of that buzzwords. And so this can be adapted to now this new field called renew or not, but this concept from DevOps to DataOps. And the three sort of key pillars of this is, the way I think about this is sort of like, the first point is how do you go from the site to the data scientist? You want smooth, fast, secure flow of information from the source to the user. But the second part is you want rapid feedback loop. Along this long chain, you need to be able to get information back to the people who are developing. Like the first thing is that the site misenters the information. How quickly can you tell them there's an error in the data? And then the last one is this concept of continuous improvement. You're all scientists here, so it seems a little bit more obvious, but you should never be satisfied with whatever pipeline you have in place. And so what are things you can do to sort of revise your processes, your pipelines? I realize this is going to be a little bit more like a management class than a data science class, but I think it's important to understand this concept. If you're going to go in a scientific career, where does that come from? So the second guiding principle is the data sharing principle, and there's many variation on it, but the one that still sticks today is the FAIR principle. So you want the data to be findable, you want it to be accessible, you want it to be interoperable, and you want it to be reusable. And really, this has been the foundation of many open science initiatives, even though FAIR does not mean open. but often they can be associated. And then, the flagship NIH-funded efforts, such the ABCD study, the Human Connectome projects, etc. So let's talk just a little bit about the smooth, fast, and secure flow. This is a traditional data flow. Okay? So here we go. You have your acquisition site. You have this big data center. You maintain these data capture systems. Of course, they're different depending on, if you enter data from a clinical interview or if you upload an MRI, you might have different databases. You dump all of this into your data processing center, some data transformation, and then you push it to your database that you want to release to the community. In this case, it's the NIMH Data Archive, which is maintained by NIMH for the research community. What was a little bit tricky in the AMP project, schizophrenia project, is that, I don't know if you remember the slide, I went over a little bit quickly, but you have these three entities. You have so orange and yellow are the research network. They recruit participants, they test participants. And the green part is the data processing analysis and coordination center, which I'm a part of. And so these are based in the US at Yale. These guys are based in Australia, in Melbourne. And so before the project started, we did not know these individuals. We did not know they were involved in this. And because of political, logistical reason, we were not absorbing at the data processing center. We're not maintaining the data capture system. So each one of these guys have different ways of capturing data. And our role is to kind of, as a separate data processing analysis center, we have to talk to the NDA and do all the QA/QC. And so that looks a little bit more complicated. You have the ProNet Hub, which is the Yale Research Network, and the Prescient Hub, which is the Melbourne Research Center. They each have their own data capture system, and then we develop tools so that we can aggregate the data on their system, because we cannot do it outside of the system for privacy reasons. We do some stripping of PII. That gets pushed to a sort of staging environment, which is owned by the US governments and NIMH, from which we can actually get the data curated and put it back in these things. And so it's a little bit complicated, but this is the reality of doing these projects. It's never as simple. And this is the PowerPoint architecture. Trust me, the real architecture is always messier than the PowerPoint architecture. And so what we've achieved through all of these complex things is we can do, so the data capture, one thing that's very important, if you can get rid of paper forms, get rid of them. So we were able to do this for everything except the IQ test, and we're still breaking our heads against the wall because they weren't scored properly, and the source document is in a drawer somewhere in Italy, and how do I check? Okay. So the MRI, the EEG, all of these recordings, we ask people to upload them within 24 hours. They sometimes do, they sometimes don't. So we need to track this. The information about the fluid biomarkers, so, the storage information, the temperature of the fridge, all of this stuff has to be put within 24 hours of sample collection. And then we aggregate this data, these little things here. This runs every 6 hours. Whatever is new here, we kind of aggregate it and push it to the next systems. So this is kind of how data has been moving since the beginning of the study in it was summer 2022 up until about last year. I don't have an updated graph, but you can have up to 10,000 variables per participant, 50 gigabyte of raw data. So this is quite a bit of data. So the second part is the quality control. So again, here, what's very important in a study like this, and I've participated in too many studies, we collect data, we collect data, we collect data. We're done collecting data, and then we open sort of the database and be like, okay, what does the data look like? And you realize, oh, this site had a problem with the MRI. They used the wrong protocol. But I have 500 MRI that I need to do some fancy post-processing on. Okay? So you want to course correct this as soon as possible. So how do you provide this feedback as quickly as possible? And you want to cover all of your domains. And so how do you do this? Do you send emails to people? How do you track that? All of these things are things you need to build in order to maintain the study. So we rely on automation whenever possible, and then we do visual inspection of EEG and MRI. But everything else is sort of automated. If there's a required field that's not entered in one of the database, the next day there's a query that gets sent to the site and says you need to fill this out. And similarly with most, if somebody should have uploaded an MRI and we don't have it, all of these things kind of get maintained. So what we're able to do is, a lot of the checks you can do, you want to do them as early as possible in the pipeline. And so when you use a data capture system and you enter a value that's not plausible, ideally you don't want to wait until I see it to tell you this data is out of range. You want your data capture system to have things in it that will say, oh, no, you can't have somebody who's a 125-year-old in this study. So we try to provide feedback within 48 hours from data capture, and this is a real challenge because if you remember my simple PowerPoint architecture, there are many, many points of failure, and so it's possible that data flow stops somewhere, and finding where might mean that the feedback is multiple days, not 48 hours. We have a general study dashboard that we update daily, and we have a bunch of other dashboards that we use for many different kind of inspections. And so we've developed a bunch of tools. I don't want to get into all of the details of this. Most of them are in our GitHub for this project. The main one is for the first pillar is this richness tool, which is this federated data aggregation. There's no person that clicks and downloads spreadsheets anywhere. We talk with API to all of these database systems to download it. We have a data visualization tool that shows the status of the project and many, many different custom pipelines. So here are some examples. This is how many participants you have at every time point. This is one of our QC tracker. You'll say, okay, for this participant at this time point, this thing has errors. It's been 81 days since we flagged it. How come you haven't resolved it? And so you sort of have all of this and then you keep track of those things. Then you have trackers for things like data quality. So when somebody visually inspects the data, we give them a score, full pass, partial pass. We don't have the data yet. The data is marked as missing or it hasn't been evaluated. So we're always trying to get these gray areas as small as possible and sort of things. And there's on and on and on the number of tools that you have to develop to analyze all of the data. Okay, so the last part of this is the data sharing. And so what we've been doing is we have a mandate for an NIMH to release the data to the community every 6 months. And this is a very tall order, and so we try to do every 6 to 9 months. That data is available as of June 25. You have about 2,100 participants for which you can download all of this data. And we have the baseline visit, the screening visit, all the way to the month 2 visit included. And our next release will be in early January 2026. And so what does that look like? Have, if you go to the NIMH data archive, there's a dedicated page. And then you have about 30 data instruments, and screening, social demographics, outcome measures, cognition, food biomarkers, and all the biomarker data. So EEG, MRI, AV. So AV, you only get transcripts at this point, but also acoustics features. You do not get the raw recording for privacy reasons. But you can get the accelerometry, GPS, screen use, etc. And these are the numbers. So this is start to get significant data. The Sykes is the main diagnostic instrument. So we have over 2,000 participants that have this, social demographic, cognition, EEG. So a nice playground for you to play with. The last step is continuous improvement. How do you implement this over 40 sites? And so what we do is, the first thing is, this is again a software thing. It's called the blameless post-mortem. Anyone heard about the blameless post-mortem? No? Okay. So the idea is if you're in a big IT company and your server goes down and everything goes to shit, pardon my French, you have a post-mortem meeting and you retrace all of the step backs, so everything that went wrong, and there are very strong rules about not blaming anyone, because at the end of the day, the rule is not to say this person did the wrong thing, it's why did this person do this, and how can we avoid having this person make this decision the next time this situation happens. And so here it's the same thing. You need to have this culture that says, you know, if you observe a problem, tell us, don't wait, because that's going to make everything worse. Their importance, and there's many things that you cannot resolve with systems. You also need cultures within organizations. We reevaluate the QC metrics, so, you know, this QC tracker I showed you, somebody is like, well, I discovered this problem with the data. Can we flag it now? So we talk and we say, okay, how we can make a new rule so that if this happens again, they'll show up on the tracker. You provide regular training, we refactor all tools all the way. The softness tool is going major. And then what we do, I'm sure many of you have participated, is this concept of hackathons. We try to have everyone get together, or a large number of people get together a couple times a year, sit in the same room, tackle a couple of problems that have been nagging us for the past 6 months, and try to find solutions for them in person. This is the scope of the project. This is just the people who were in New York 2 years ago. There's about 150 main scientists. That's not accounting for all of the research assistants and all of the study staff technicians. So where do we go from there? We're about done with recruitment. This summer, the first papers are coming up with the initial baseline findings. We'll hopefully release the final baseline through month 2 data set. This is going to go all the way through February 2028 by the time we're done analyzing this data. So this is a major endeavor that started in 2020, so an 8-year project for this. Another exciting part of this project is the NIMH released more funds than their partners, and now we're going to start to do clinical trials too. And so we're doing this thing called proof of principle clinical trials, which is, you know, you have a compound, some medicine, you've done all the safety measures, you've done some initial testing that shows it may have some things, and then the proof of principle, it's really about, okay, if I give this medicine, I have the assumption that it's going to change these biomarkers. Let's verify this. It's much more that than saying this person is getting better. So trying to understand how the drug works. And so we're using the same platform, the same tool, the same data engineering with the added constraint of a clinical trial randomization, FDA, some legalese that I won't bore you with. Okay, so I think I'm getting towards the end. I want to conclude my talk with sort of my final take-home message, share it. And so how many of you know this paper? Variability analysis of a single neuroimaging data set by many teams. Okay. Anybody care to volunteer what this paper is about?
They gave 70 different teams the same neuroimaging data, told them to analyze, I think, 9 hypotheses, and then they found out that no one agreed with each other.
Yeah. It's not quite as no one, but rarely more than 50% of the people agreed on a particular hypothesis, right? Why is that?
Different methods, different pipelines.
Even the same pipeline, just different parameters. Sometimes even the same method, and somebody who's no longer at Concordia, please come back to work on this. The exact same tool. If I run on a Linux machine or a Mac architecture, I'm going to have variations because the underlying libraries may be different, and it could be a very minimal impact, or it could have a real impact. And so, you know, it's important to validate and share complex analysis, workflow demonstrate. And so this is one problem, why you need to share it. But it's not enough to just do, okay, here's open science, go my GitHub, you have it. Yeah. How many of you have tried to run something that you downloaded from GitHub and it didn't work? Yeah, not 100%. Okay. And so this is the next bit, right? This is also a software engineering concept, which is this technology adoption that you have these called the Morse chasm, where you have this curve of adoption. You have the early adopters. They don't care if it works halfway. They'll just want to try the new toy. We'll all go through this phase. Then the early adopters are there. And at some point, you have to convince the majority to use those tools. How do you do this? How you cross that bridge is not simple. And at a minimum, you need to have software that's well engineered, well documented, easy to use. And these are not simple things to do. And so we have to have quantitative and qualitative. You see, when you do reproducibility, you need to know if your data is good, if your outputs are good. And so without getting into details, I spent a lot of time on this. And so we have a lot of neuroimaging pipelines that we put on GitHub, the things that we think are useful to the community. We usually go to the effort of refactoring it, documenting it, having manuals. And then we also share data. So the AMSKits is, of course, the biggest project, but the early side, we have another one, which is the raw data. All of these annotated training data we also share. And I think something that people do a lot less that to me is equally important is your protocol, your standard operating procedure. Right? When we do the AMSKits project, you say, here's the MRI, but actually you're like, okay, you get the patient in, you give them earplugs, you tell them what to do, you do all of the checks. And so some of this are very standard, everyone knows them, but others may have a very strong impact on the data. If you do the cognition at the end of a 2-day testing, you're not going to get the same performance than if it's the first thing you do in your protocol. So all of these, if you can share it, do it. And so we use GitHub, we use NDM mostly for data, not only, and Zenodo is one of the platform I like best for just kind of general sharing. So in conclusion, unless you have Canadian requirements, then you use Borealis. So in conclusion, I think to go all the way back to my first thing, the commonly used neuroimaging pipelines, they're not set in stone. They have limitations. There's a lot of fun work to do there. And so if that interests you, just go for it. There's lots of things still to be done at this level. We need plentiful and high quality data to answer important clinical questions, and it takes a tremendous amount of resources to collect high quality data. And so, 2,000 subjects, $100 million, that's $40,000 or $50,000 per participant. Of course, this is a caricature, I'm exaggerating, but half of that amount is not an exaggeration, really, to get all the expertise. And so it's very valuable. You got to share it. You can't just keep it in your drawer. I hope I convince you that data science needs data engineering and software engineering principles can be adapted to have more reliable and reproducible science. And, sharing is critical to advancing science. I think we can all agree with this. But if you're ever going to have your own lab or participate in the lab, do not underestimate the amount of work it takes to do that sharing. And that is not usually rewarded in your paper, but I think it's as important as your mission as a scientist to do today, to participate in that effort, to actually make meaningful impact as opposed to another line on your CV. So thank you very much for your attention. If you have any questions, I'd be happy. Any questions? Yeah.
Yes, thank you. Thank you very much for your presentation. I really like the part about tool development. I don't work in brain imaging, but I work in another sort of image analysis, and tool development is something that I do a lot. And so I was wondering, in this project, for the tool development, who is it that works on it? Are there hired engineers? Is it students? Is it research assistants? Is it a mix of everything? And what do those teams look like?
So it's the latter. So for the AMP schizophrenia projects, There are 2 data engineers / bioinformaticians. There are, let's say, 2 or 3 postdocs. Some are more technical, some are more sort of clinical neuroscience-y. That's just the data management core. And then there's 2 or 3 research assistants, all with, so by research assistant, because I know that definition can vary. We're talking about somebody who finished their undergraduate degree and is taking a couple years in a lab to learn about research before they decide what to do with grad school. So overall, you know, this is the team. The core team is about 5 to 6 people. But then around this, for each domain, in an ideal world, we have a technical engineer-like person that understands the domain and a domain expert that need not be an engineer, but needs to know that this electrode on the EEG recording needs to look like this. So.
Okay, thank you.
Yeah, so you mentioned how your imaging was having issues with sometimes doing things that were like not biologically possible, and you overcame the challenge with some different units. How did you overcome the challenge?
Which challenge?
Just like basically the segmentation was kind of like going from like left hemisphere into the right hemisphere, just those biological impossibilities. How did you update your machine learning pipelines to really prevent biological or physical impossibilities?
So I would say each one of these sub-tiny problems could be a PhD thesis on its own. Okay.
I know.
And so
I know.
in the context of these cortical surfaces,
Yeah.
Okay, the best way to typically the way this works, let's go through the for workflow just for hand wavy PowerPoint architecture. So you get a brain MRI. It's going to identify the white matter from the rest, get the contour of the white matter. And then from this, it's going to sort of grow or try to estimate the PL surface. And then it does this for the left hemisphere, it does this for the right hemisphere, independently. So you do white matter, PL, white matter, PL. Okay. There is very little awareness of what's happening on the other side. Okay? And so the way we're doing this without talking about the deep learning is saying, I give you 4 initial surfaces, just blobs. It could be, to simplify this, I have a sphere inside a sphere inside another sphere. They don't touch, they have all of the right properties that we want. And now I'm going to deform my space using a transformation called diffeomorphism. Does that ring a bell, diffeomorphism? Homeomorphism? Okay.
Yeah.
So it's just something that just means that homeomorphism is a transformation that does not change the topology of objects. So if you have a sphere, it can become a cube, right? But it will not become a donut. You cannot cut, you cannot add a handle. So you can deform it however you want. And so that's what we do. Also, if 2 objects are next to each other, the diffeomorphism or homeomorphism does not allow you to have things that end up intersecting each other. And so people have used diffeomorphism in the past, but the idea in this particular case was just saying, if I start with 4 surfaces that are well formed, I only allow diffeomorphic transformations, then these things are theoretically guaranteed not to intersect, not to do these 4 behaviors. So that's one example of how we deal with that.
What's the paper's name?
It's here. You can have the slide.
Yeah.
I'll provide I'll give the PDF of the slide, but it's going to be presented in this at McKay, the SHAPE McKay workshop. And there's an archive paper. If you put these 3 in archive, I think that's the only paper. Yeah.
This isn't really to your main point, but I'm just kind of curious. How much expertise was required when you have the manual checking of the MRI and EEG?
So manual checking.
When you have someone physically view them, like how much prior knowledge do they need in training?
It's fairly quick. So in my former lab, we would have these research assistants come in all the time. I would say one of the first things they learn is this. So we have a manual, and then you have a training procedure. You have these brains that have different scores, and then you say, okay, if you observe this, that means this score. And then they go through, and then once they get to similar scores, then what we have as our stick. So I would say, within a month, and then within a few months, you're an expert for the things that you see. And then once in a while, you have a brain that looks very weird, and then you go talk to the neurologist.
Maybe one question on the, we haven't talked much on the difference between like sensitive versus non-sensitive data. Maybe all the data have the same kind of sensitivity and privacy requirement in that project. I was a bit curious about how that was handled in the schizophrenia project. And my second question is a bit more open, like, let's say in 2028, things are winding down and NIH hasn't got any much money anymore. Like, what is happening to the infrastructure? And what do you have any kind of a plan or thinking about what's next and how to?
Yeah, so there's two plans. The good news is, barring any new news from south of the border, the NIMH data archive is there to stay. So that data is there for everyone to consume for eternity or as long as it's relevant. So that's step number one. As for all of the infrastructure, we have plans. So right now, it's kind of drinking out of a fire hose to make sure that all of this works every day. But we do have plans to take the most important components of this data engineering tool and make them usable for your own study. So in particular, things like this Loch-Ness aggregation tool, I think is a great Swiss army knife to many studies, because having some really lightweight server that can talk to all different kinds of data, standard data capture system, and automatically download it, organize it, track it from the database is something we want to release. But today, it's not in the releasables category. It's download at your own risk and adapt as you wish category.
Any other question for Sylvain? If not, thanks again.
Thank you so much. Sorry.